Hello, everyone. My name is Kimai Benjamin, and welcome to today's lesson on receptor tyrosine kinases and their role in cancer. We'll explore how these powerful signaling molecules help regulate vital cellular processes and what happens when their control is lost. Understanding RTKs is essential not just in cell biology but also in oncology and personalized medicine. Receptor tyrosine kinases, or RTKs, are a large family of membrane-spanning receptors that transmit signals from extracellular ligands to intracellular pathways. They regulate processes like cell growth, differentiation, and metabolism. Also, they regulate survival. This regulation of RTKs is implicated in many diseases, especially cancer. So, let's unpack what RTKs are in a little more detail. They are a specific type of transmembrane receptor that plays a central role in how cells receive and respond to external signals, especially growth signals. RTKs are called receptors because they sit in the plasma membrane and receive signals from the outside of the cell. These signals are typically in the form of ligands like growth factors. The TK or tyrosine kinases is because they have an enzymatic domain on the inside of the membrane. This domain can transfer phosphate groups to tyrosine residues on proteins. So how do they function? When a ligand binds to the extracellular part of an RTK, the receptor undergoes a process called dimerization. It pairs up with another RTK. This then activates the intracellular kinase domains. They then autophosphorylate and add phosphate groups to each other's tyrosine residues. But why does this matter? The phosphorylated tyrosine acts like landing pads for other signaling proteins. This then triggers cascades that regulate cell division, survival, metabolism, migration, and differentiation. In other words, they help the cell make major decisions. But Mr. Benjamin, how is this relevant? When RTKs work properly, they're essential for normal growth and development. But when something goes wrong with them, like a mutation or the receptor being overproduced, it can lead to uncontrolled cell division and therefore cancer. That's why understanding RTKs is crucial in cancer biology. There are 58 known human RTKs. They are grouped into 20 subfamilies that are based on structural similarities. So now let's talk about structure of RTKs. So we hit on it a little last slide, but in general, RTKs share a common structure, an extracellular ligand binding domain, a single-pass transmembrane domain, and the intracellular domain containing tyrosine kinase activity. Once activated, the intracellular domain autophosphorylates on specific tyrosines, which creates docking sites for downstream signaling proteins. Now let's take a closer look at how RTKs actually work. What's happening at the molecular level once a signal is received? This mechanism is what makes RTKs powerful regulators of cell behavior. And it is also what makes them dangerous when things go wrong. So step one is ligand binding. RTKs are initially inactive and exist as single polypeptide chains, also known as monomers. These polypeptide chains are in the cell membrane. When a specific signaling molecule or ligand binds to the extracellular domain of the RTK, it induces a conformational change. An example of this would be ligands like the epidermal growth factor, otherwise known as EGF, or insulin, binding to their respective receptors, fitting into them like a key in a lock. Step two is the dimerization. This ligand binding causes two RTK monomers to then pair up in a process called dimerization. Depending on the receptor, this can be ligand mediated, where the ligand helps to link the two receptors, or receptor mediated, where the ligand causes a shape change that allows the receptors to interact. This process brings the intracellular tyrosine kinase domains close enough to each other to become catalytically active. Step 3 is autophosphorylation. Once the kinase domains are aligned, they phosphorylate specific tyrosine residues on each other's intracellular tails. This is called autophosphorylation and is a crucial step. It turns the receptor into a platform for signaling molecules to bind. These phosphorylated tyrosines act like molecular zip codes. Each one recruits different intracellular proteins that recognize the phosphotyrosine sequences through SH2 or PTB domains. The final step, step 4, is the signal transduction or activation of downstream signaling pathways. Once these adapter proteins are recruited, they initiate a cascade of intracellular signaling pathways. Two of the most important ones are the RAS MAPK pathway and the P13K AKT pathway, which we will discuss later in this video. The RAS MAPK pathway leads to the activation of transcription factors that regulate cell proliferation and differentiation. And the PI3K AKT pathway promotes cell survival, growth, and metabolism. Both of these pathways are tightly regulated under normal conditions, but If RTK activity is not controlled, these pathways can drive cancerous behaviors like unchecked division and resistance to apoptosis. So to summarize, RTK activation follows a clear sequence: ligand binding, dimerization, autophosphorylation, and initiation of downstream signaling. Each step is essential, and when dysregulated, any part of this mechanism can contribute to tumor formation or progression. So, as I stated last slide, when RTKs are activated by ligand-like growth factors, they autophosphorylate, which creates docking sites for specific adapter proteins. These adapters then trigger several major signaling pathways. So now we're going to go into deeper detail about these pathways. As stated in the last video, we're going to first talk about RAS and the MAPK pathway, which promotes cell proliferation. RAS is a small GTPase that acts as a molecular switch. Once RTKs activate RAS via an adapter protein called GRB2 and a GEF called SOS, RAS triggers a cascade invoking RAF, MEK, and ERK. This is known as the MAP kinase cascade. This pathway ultimately leads to the activation of transcription factors, the nucleus that promotes gene responsible for cell division and growth. RAS mutations are among the most common in cancers, especially pancreatic, colorectal, and lung cancers. When RAS is stuck in the on position, it can lead to uncontrolled proliferation. The second pathway I want to speak about is the PI3K AKT pathway, which promotes cell survival and metabolism. This pathway is critical for preventing apoptosis and promoting cell metabolism and survival. PI3K is activated directly or indirectly by RTKs and converts PIP2 into PIP3 in the membrane. This attracts AKT, also known as protein kinase B, to the membrane where it becomes activated. AKT then goes on to activate proteins that promote survival and growth and inhibit pro-apoptic factors. Overactivation of this pathway is common in cancers, especially breast and prostate cancer. Additionally, mutations or loss of the tumor suppressor PTEN, which reverses PI3K's action, can also cause hyperactivation of AKT signaling. The last pathway that I want to speak about is JAK-STAT, which regulates immune response and cell differentiation. This pathway is often linked to cytokine signaling, but also involves RTKs in some cases. Janus kinases, otherwise known as JAKs, associate with certain receptors and become activated, which in turn phosphorylates STAT proteins, or signal transducers and activators of transcription. STATs dimerize and translocate to the nucleus to regulate genes involved in immune responses, cell growth, and differentiation. This regulation of this pathway can contribute to hematological malignancies like leukemia and lymphoma. Each of these pathways has a distinct role, but they can also overlap and interact. Their balance is critical. If even one becomes overactive, it can tip the cell toward tumorigenesis. This is why targeting these downstream pathways has become a key strategy in cancer therapy, which we'll speak about later in the video. So as we talked about earlier, RTKs can regulate many things such as cell proliferation, cell survival, differentiation, metabolism, and migration. So let's talk about what happens when RTKs are dysregulated. RTK signaling going wrong is a central theme in cancer. First, RTKs can be overexpressed. This means that the cell produces too many of them. This makes the cell overly sensitive to growth factors and it responds even to low signals. A great example of this is the HER2 overexpression in some types of breast cancer, which we will go into further detail later in the video. A second mutation in RTKs can also cause them to be permanently active, even without a ligand. These mutations often affect the kinase domain or transmembrane region. One common example of this is EGFR mutations in lung cancer, which can lead to continuous cell division. We'll further break this down later in the video. Third, RTKs can undergo ligand-independent dimerization, where they pair up and activate without a signal, especially in cases of gene fusions or structural rearrangements. Finally, cancer can arise when mechanisms that normally shut down RTK signaling fail. For example, in the CBL ubiquitin ligase, which normally tags RTKs for degradation, may be mutated or underexpressed, which leads to sustained signaling. All of these changes contribute to the classic hallmarks of cancer: uncontrolled proliferation, evasion of apoptosis, sustained angiogenesis, and more. So like I stated in the last slide, here are some examples of RTKs in cancer. HER2 in breast cancer, we will dive deeper in this video. EGFR in lung cancer, we'll talk about that in more detail as well. But c-KIT and GIST is an example of cancer that we will not talk more about, but you can research upon your own. We can also detail how there are other cancers associated with RTKs, such as VEGFR, which promotes angiogenesis in tumors, PDGFR, which is involved in glioblastomas and leukemias, ALK, which deals with gene fusions that drive some lung cancers, and MET, which is overexpressed or mutated in several tumors. All of these are targets of ongoing drug development. I challenge you to pick one and research it. So now let's look at the real world example of HER2 in breast cancer. HER2 is a member of the ERBB family of RTKs. In around 15 to 20% of breast cancers, the HER2 gene is amplified, which causes overexpression of the HER2 receptor on the cell surface. This means that about 1 in 5 women who experience breast cancer has this issue. This overexpression leads to a constant dimerization and activation of the RTK, even without ligand binding, resulting in continuous signaling through the MAPK and PI3K AKT pathways. This results in rapid cell growth, survival, and tumor progression. Clinically, we use tests like IHC and fluorescence and FISH to determine HER2 status in patients. Targeted therapies like Herceptin or are monoclonal antibodies that bind to HER2 and prevent dimerization. They also recruit immune cells to kill the cancer cells. HER2 targeted therapy has dramatically improved survival rates in HER2 breast cancer. This case perfectly illustrates how understanding RTKs can directly lead to targeted, effective treatment. Another key example of RTK involvement in cancer is EGFR in non-small cell lung cancer. EGFR mutations are particularly common in Asian populations and non-smokers with non-small cell lung cancer, otherwise known as NSCLC. These mutations often involve exon 19 deletions or L858R point mutations in the kinase domain, which leads to constitutive activation of the receptor. Because the receptor is always on, cells proliferate uncontrollably. But here's the good news. This discovery led to the development of targeted therapies like erlotinib and gefitinib. These drugs are tyrosine kinase inhibitors, or otherwise known as TKIs. They bind to the ATP pocket of the mutated EGFR, shutting down its activity. However, resistance often develops, such as the T790M mutation, which blocks drug binding. To address this, third generation inhibitors like osimertinib were developed. So here are some targeted therapies for RTKs. Monoclonal antibodies block receptors. TKIs block kinase activity. These treatments are more selective than traditional chemotherapy and often have fewer side effects. I challenge you to research some resistant developments in these inhibitors and to find some off target effects. Resistance to TKIs is a major challenge, and it may come about due to secondary mutations, activation of alternative pathways, and RTK amplification, Overcoming the resistance requires combination therapies or new inhibitors targeting resistant forms of the RTKs. Because of this, we have emerging research and future directions and newly emerging therapies. For example, personalized medicine, which is based on genetic profiling and diagnostics, relies on accurate tests. Tumor biopsy and genetic profiling can reveal mutations in RTKs. For example, HER2 status determines eligibility for the inhibitors that target HER2. EGFR mutations inform treatment with TKIs to ensure that patients get the most effective and least toxic treatment possible. There are also new frontiers such as bispecific antibodies that target two RTKs, antibody drug conjugates targeting RTK degradation pathways, and using RTK signaling to boost immunotherapy. I challenge you to complete this activity. We are now finishing the lesson, so I want you to review a patient case with ESGFR mutation, identify which RTKs are involved, propose a treatment plan, and discuss resistant mechanisms. Key points to remember are that RTKs are critical in cell signaling. Dysregulation of RTKs can lead to cancer, and targeted therapies can improve outcomes. Are there any questions? If not, thank you for watching this video. This has been Kimani Benjamin and receptor tyrosine kinases in cancer. Thank you.